maybe you can moderate your question. Good afternoon. Welcome to Oncology Yale Cancer Center Grand Rounds. My pleasure to be the host today. We have two speakers, as is our usual format. Our first, a more basic science talk, and our second, we actually have a special visitor all the way from Ireland to tell us a little bit about what's going on there. So for the first talk, it's really my pleasure to introduce Dr. Anthoy Koleske, who's well known to most of us, who's a professor of molecular biophysics and biochemistry, MB&B as it's known. And he's going to talk to us about the mechanism of assembly of an invasion control switch in breast cancer. Tony.
Okay, great. Can everyone hear me? Great. Thank you all for coming. So in starting out, I want to remind you actually of some statistics that you're probably all quite actually familiar with. So it's been estimated there's something like 250,000 of cases of newly diagnosed breast cancer every year in the United States. And about 75% of these cases are invasive carcinomas. And of course, I'll talk a little bit more about this, but invasion is really the first step in metastasis. And it's complications from metastasis that are the primary cause of mortality from breast cancer. And so trying to understand this and potentially target it is an important goal. So breast cancer cells escape or initially become invasive by penetrating the basement membrane that surrounds the tumor. And they actually utilize very specialized structures to do this. They're called Invasive Podia, and I'll talk more about these. And Invasive Podia actually help them digest and penetrate these basement membranes. Now, they seem to migrate in lockstep, though, with tumor-associated cells, such as macrophages. And what macrophages do are they provide a chemokine, a growth factor called EGF, and that seems to be a particularly potent stimulator of these structures, these Invasive Podia, that I'm going to tell you about today. So this is a diagram of an Invasive Podium forming at the bottom of the cell. So the cell is sitting up here, and you're sitting on top of basement membrane, and it's poking down into the matrix. What you'll notice is that there's a collection of multicolored proteins here, many of which are regulators of the actin cytoskeleton. And their job is actually to nucleate the formation of these actin filaments, which elongate, and they actually provide a pushing force that drives the Invasive Podium down into the matrix. And at its tip, these Invasive Podia can secrete matrix metalloproteinases, which are specialized enzymes which help degrade the matrix. And of course, the idea here is that you push out with this main apparatus and you degrade at the tip. And so thereby you can very efficiently penetrate these membranes. And this sort of just shows you, this slide shows you this is a breast cancer cell sitting on top of an artificial reconstituted basement membrane. And you can see the actin cytoskeleton lit up here in green. What you'll notice here is this concentration of green penetrating, and there's a slight breach in the basement membrane. Over time, that breach becomes more significant until, in fact, the Invasive Podium can essentially drive a wedge, and the cell will actually crawl through. Now, we can study Invasive Podia by virtue of their ability to degrade matrix, and the way we do this is to plate cells. These cells are this cancer cell expressing a green fluorescent protein actin, so you'll be able to see the actin cytoskeleton here. We can plate them on fluorescently labeled extracellular matrix. So here, this brightness is that what you're looking at is a full-blown brightness of the extracellular matrix. And what we're going to see as this movie plays, you're looking down on top of the cell. You're going to see an Invasive Podium form right here and stabilize. And what you can hopefully see is that coincident with that stabilization, you begin to see degradation of the matrix, and you get a clearance zone there, which appears as a dark spot on the fluorescent background. Now I'll show you examples then where we use this assay to sort of quantitate the efficiency of this degradation process. work from a number of people in the field. I think most notably one of our collaborators on this project, John Kondilis, has established that these Invasive Podia actually form in stages. So initially you can observe clusters of these actin regulatory proteins I told you about, and you can see them sitting there. They're sitting just below the surface, ready to go. But then something happens, and I'll tell you later that that trigger seems to be EGF. And you get a polymerization, a burst of actin polymerization. And that seems to stabilize these structures and allow them to mature and begin to degrade the matrix. And so we wondered whether or not we could somehow figure out ways. This is my expertise is actually in trying to figure out how actin polymerization is regulated by external stimuli. We reasoned that we might be able to block this step and try to see if that could block Invasive Podia formation. And even better, could we block that step and try to prevent breast cancer invasion either in vitro or ultimately in vivo? And so my interest, our entry point into this is in our interest in studying a class of enzymes called Abel family kinases. And you're probably familiar with Abel because of its association. It's mutated in a number of leukemias, notably chronic myelogenous leukemia. There's another enzyme that's very similar called Abel2. We call it ARG. And the job of these kinases is to act downstream of either growth factor or integrin receptors and transduce those signals into changes in cell structure. So what they do is they target various substrates. They also can actually engage. They have a scaffolding function at their C-terminus. They can engage that. And in engaging these substrates and effectors, they change the structure of the actin cytoskeleton. And our work and work of others in the field has shown that these are really powerful coordinators of changes in cell shape and cell movement. And just to give you one illustration of this, this is a cell that's expressing its endogenous copy of ARG has been deleted. Both copies have been deleted, and it's been replaced by a fluorescent protein called fluorescent ARG fusion. And so we can monitor what's happening to the cell and also what's happening to ARG. And what you can see is these cells that are slightly overexpressing ARG, when we put them down on matrix, they undergo these very dynamic protrusions out at their periphery. And everywhere we see one of these protrusions, we see a concentration of ARG. Okay, so that was our initial question: is how is ARG talking to the actin cytoskeleton to produce these changes in protrusions? So we know that it was engaging somehow with the integrin receptors, and that's a story I don't have much time to tell you about. But the question was, how is it talking to the ARP2/3 complex, which is the main nucleator of actin polymerization in these structures? And we hit upon a link, and the link was a substrate we identified of ARG called cortactin. And cortactin has a nice central domain that binds to F-actin, but what it has is it has a module here that can plug into the ARP2/3 complex and activate it. And it also has a C-terminal domain that can plug into another protein called NWASP, which is also a very potent activator of the ARP2/3 complex. So it really has two domains that can plug in and superactivate the ARP2/3 complex. And we were able to show actually that ARG could phosphorylate this very well at a number of sites. This is, if there's any biochemistry geeks like me in the audience, this is sort of a phosphorylation curve where you can measure the Michaelis constant for this. To simplify this, what this tells you is that ARG is actually recognizing cortactin at something like 1 part in 100 billion, which is a pretty good enzyme-substrate relationship. We went on to show that ARG phosphorylates 3 sites. We actually know now that only 2 of them are important, and I'll talk about those a little bit more. Now, cortactin is what moved us from beyond sort of these fibroblast cells to working on breast cancer. Because in looking at the literature, we knew that the locus that contains cortactin is amplified in a variety of invasive cancers, including breast cancer. And in some cases, more cortactin protein seems to be associated with advanced stages of the disease and poor patient prognosis. And we went on to show that cortactin phosphorylation at 2 sites, so 421 and 466, seemed to be really important for the function of Invasive Podia, matrix degradation, and cellular invasiveness if we engineered breast cancer cells with these manipulations. And so we thought that phosphorylation of cortactin is this key trigger to trigger invasion. Could we somehow disrupt that process and prevent this step and prevent Invasive actually from forming? So the first question was whether or not we could identify what was responsible for phosphorylating cortactin. And here we took a knockdown approach. So these are, all of the work I'm going to talk about in cells is in MDA, MB231 cells, which is a very invasive breast cancer, triple negative breast cancer cell line that many people use. Here we show that we can very efficiently knock down ARG or the closely related ABL or another non-receptor tyrosine kinase called SRC. And I'm not going to talk about SRC, but for everything I talk about today, I'm going to tell you, we published on this, SRC seems to be a collaborator of ARG. It seems to do something very specific to activating ARG, and we could talk about that more offline. So the first question was, in these knockdown cells, do we diminish cortactin phosphorylation? I remind you that EGF is this potent stimulator of cortactin, of both cortactin phosphorylation and Invasive function, and in fact, when we treat these cells with EGF, 3 minutes later, if we measure tyrosine phosphorylation of cortactin using a phosphotyrosine antibody, we see a very robust increase in phosphorylation in these small spots that correspond to these invadopodial precursors. In contrast, in ARG knockout cells, we don't really see that response. And in fact, if we quantitate this over thousands of these dots, what we find is that there's something like a threefold increase in phosphorylation of cortactin in control cells. We completely wipe that out when we wipe out ARG, whereas if we knock down ABL, there's absolutely no effect. And we now know that this is because ARG itself localizes to Invasive, but ABL does not. It's expressed in these cells, but it seems to be everywhere else except for Invasive. The cortactin phosphorylation is actually a trigger, as I said, for actin polymerization. And here we've used an assay where we can mildly penetrate the cells with detergent and float in a little bit of actin. And that actin will actually get incorporated into all the new actin filaments that are forming. And so the brightness of the spot actually corresponds to the magnitude of the actin polymerization response. So again, here, EGF stimulates, both you see more spots and you also see larger spots, okay, in the EGF-treated cells. You also see this in the ABL-treated cells. Remember, ABL doesn't have any inhibitory effect on this. But in the ARG knockdown cells, we don't really see any effect at all. And here again is the quantitation. We see something like a 2 to 2.5-fold increase in the amount of actin forming at these invadopodial precursors. Whereas if we knock this down, if we knock down ABL, there's no effect. If we knock down ARG, there's a pronounced effect. We can also measure matrix degradation. I showed you a movie of just a single little Invasive forming and a clearance zone resulting. If we let these things go for a couple hours, you actually see lots of black spots forming. And we can put a mask on this to quantify this. And what you can see is in the ARG knockdown, there's a lot less clearance zone. And in fact, we see a significant reduction in clearance of the ARG knockdown cells. And the ABL, again, are largely unaffected as the control. And finally, with the cell-based assays, we wanted to ask whether or not this compromises the ability of these cells to traverse, to penetrate matrix barriers. And so we use these tissue culture insets that have small pores in them such that cells can crawl through, except that we coat them on the outside with matrix. So the only way the cell can get to the bottom well here is to actually digest the matrix that's above these things and then crawl through. And if you're on the receiving end on the underside, this is a scanning EM of what that cell crawling through would look like. And in fact, we showed that the knockdown of ARG actually significantly compromises the ability of these cells to crawl through matrix and penetrate these barriers. So I want to come back to a point here, just to sort of tell you where we're going with this sort of biochemical and in vitro studies. And that's really focusing on this issue that cortactin phosphorylation is a major trigger for these Invasive to form. And what we've done is we've enlisted Bruce Mayer and Kazuya Machida at UConn in a high-throughput screen. And as you probably know, tyrosine phosphorylations, when they're on proteins, are actually a recognition sequence. They're the anchor for protein modules called SH2 domains that are found in many signaling proteins. And so what we did is to simply spot out either non-phosphorylated cortactin, phosphorylated cortactin, or positive controls. These are, I think, EGF or pervanidate-treated cells. They have a lot of phosphotyrosine in them, and negative controls, which are phosphatated-treated extract. And so what we do is we take these disks and then we screen them against every SH2 domain in the human proteome. And so what you can see is if we were to do this with two different SH2 domains here, this SH2 domain only binds, both of them bind to the positive control here, these two spots in the middle. So you can see the signal there. Whereas one of them, the ARG SH2 domain, only binds to the phosphocortactin. Okay? And we actually used this and we came up with a lot of interesting things. And we're able to estimate the relative affinities by monitoring the concentration dependence of this screen. And somewhat gratifyingly, ARG, our favorite molecule, actually came up at the top of this list as the highest affinity binder. Of course, we know that's an important interactor. I'm going to tell you about one of the other candidates we pulled out just briefly, just as a sort of an example of the fact that we think this has been very fruitful. So VAV2 is actually a GEF. It's a protein. It's a guanine nucleotide exchange factor. So its job is to take a small row family GTPase and activate it. It rips out the side of it, and GDP falls out, and GTP, which is much higher in concentration, gets in there, and it activates this. And this has a different conformation than this by virtue of binding GTP, and it can engage various downstream events, and you're off to the races. Now, you probably know that there are multiple row family GTPases which exert different influences on different actin-based structures. And so the first thing we needed to do, this is Brian Rosenberg, needed to figure out what it was targeting. And Brian went on to show here, doing a pull down, what he's used as beads, just GST beads alone, or GST beads coupled to RORAC or CDC42 that are mutants, and they're mutants that mimic this GDP-bound form. So they should bind to the GEF with very high affinity. And what you can see is that this GEF seems to have great selectivity for RAC. And so that's good. That means Brian's going to have a thesis. And he's off to the races. One of the first things he did was to knock down VAV2. You can see he's done so very effectively here. And at least our preliminary data suggests that that also completely wipes out the ability of these cells to degrade matrix. And this is quantitated down here. So he's trying to follow up on this and wrap this up right now. Now, in the end here, I want to turn to what the relevance of these studies might be to breast cancer spread in vivo. And here we engaged in a collaboration with John Kondilis' lab, namely Tanya Patsialu and Hava Gilhen from my lab, who's now moved on. And the idea was to engineer cells where we've manipulated ARG and also labeled them green and use them in a xenograft model, a skid mouse xenograft model, and ask how, if we compromise invadopodial function, how this affects tumor spread in vivo. So here you can't really use siRNA because you want it to be permanent. So Hava and Tanya used shRNA to ARG to knock it down. And just to confirm things, to make sure this had the same effect that the siRNA had, they measured the ability of these cells to invade through matrix. You remember that assay I told you about where we have the barrier above the porous membrane, and we ask whether they go through. Well, if we knock down ARG using this technique, they also have problems with that. And so now we can take the cells and inject them into the mouse memory fat pad orthotopically, and we wait a variety of time, and what you can see, of course, is that tumors form. And when the tumors are about 1 to 1.2 centimeters, cubic centimeters, we begin to score invasion, intravasation, metastasis. And it's nice to have these green cells because you can easily identify them and be very quantitative about that. And so the first thing Tanya and Hava did was to look in the blood. And in contrast to the control cells, what they found is both of the different knockdown lines seem to be significantly reduced in their ability to find their way into the blood. This is just in blood that was isolated from from the mouse. We also went on then to ask whether or not this particular cell line likes to metastasize to the lungs. And so you can see these metastases just doing basic histology. So we did both this, and I should say we also looked at micromets by just looking for green cells. We can talk about that if you have questions. But what we found, at least at a gross level, is that these cells really don't form mets anywhere near as well as the wild-type cells. So that's great. That sort of is perfectly in line with what we were thinking, and we should have just left it at that. It turns out that we got a real surprise from these. So in the orange knockdown tumors, in contrast to the other tumors, they're absolutely huge. Okay, so you can see, you can compare scale bars. So the orange knockdown 2 are even bigger than the orange knockdown 1. And if we look in these tumors for a marker of proliferative cells, what we find is that that's greatly increased in these knockdown cells. And I should point out this was completely unanticipated from our work. And if we look here, we did this sort of in a very directed subset of genes that either regulate proliferation or regulate invasion. And what we found is that the knockdown in both knockdown lines relative to controls, the genes that were involved in proliferation were all increased. And this collection of genes that we know are involved in invasion, in an invasion signature, are decreased. Now, consistent with that, we went on and actually just measured things like RAS activity. We found actually that RAS levels were actually increased, as was RAS activity. And we can look at things like MAP kinase activity. That's also increased, even in staining in the tumors. If you stain in the tumors, it appears that the activity of this pro-mitogenic pathway is actually upregulated as well. So I think the ongoing challenges here, this was a surprise, but really to try to understand how the loss of ARG increases proliferative potential. As you probably know, there are very good inhibitors of these Abel family kinases, and we're just starting out now to test whether or not Gleevec or dasatinib, which both, I should point out, they both work equally well on Abel and ARG. We're trying to see whether or not that can influence the spread of tumors in vivo. And could that somehow be an adjuvant for therapy in the right application? And I would leave that to the judgment, I think, of the clinicians here. Can we find other targets or inhibitors to block Invasive Podia? I showed you at least one promising candidate on the list today. There are others on that list that we're quite optimistic about. And then I think finally, and this is something I have to thank really the program leaders at Yale for, because we didn't know how to do this, and they matched us with some people that did. Do we see correlates of these biochemical changes in the clinic, and are any of these markers useful for staging and predicting outcome? Now, we don't have an answer here, but as I alluded to, the program leaders put us in touch with Kim Jo-hung and David Rim, and student Jin Meng, who is beginning to look at this. And what you're looking at here is a tumor sample that's been analyzed using David's Aqua technology, staining for cortactin. And what you can see, hopefully pretty clearly, is that these are all different samples on a tissue microarray. And you can see that there are different, very clearly different signals and different scores. And so we're interested in things like cortactin. We also have antibodies that recognize different covalent states of that we think represent distinct biochemical stages in this process, in this transformation as well. So with that, I really want to acknowledge the people who did the work. So Hava Gilhen did all the animal work in my lab, is now at the new medical school at Bar Ilan in Israel, a new assistant professor. Stacy McGrath did some foundational work on the cortactin that I didn't really have time to talk about. Brian Rosenberg's MD PhD student working on VAV2. Titus and Wei Zi and I are engaged in a long-term collaboration, actually with Stacy, to really we've done some very nice structural work on what cortactin looks like, and especially what the cortactin-ARG binding interface looks like, because we think that might be a pretty valuable target for disruption. I talked about Tanya's contribution to this project and our long-standing collaboration with John Condilis. And here's a picture of David Rim. I couldn't find pictures of Kim and Jin that would appear, but we also appreciate their help and are looking forward to continuing that collaboration. And then Kazuya Machida and Bruce Mayer at UConn were very helpful in the SH2 domain screening. And then Chris Mader and Matt Oser were from my lab and John's lab, respectively, and really got this whole thing started about 5 years ago. So thank you for your attention. I'll take whatever questions you might have.
Thanks a lot for a wonderful talk. Good to see you had good use of Yale YCC pilot plugs
Yes,
there.
yes.
Do we have any questions or comments? Yes, David.
If you looked at Abel activity when you knock down ART,
Whether it goes up. We know it doesn't go up at the protein level. We haven't looked at the kinase activity level, but
I'm just wondering whether it's a compensatory response.
yeah,
Does imatinib have any impact after you knock
imatinib have an effect on.
down on the enhanced proliferation of these cells?
Oh, interesting. We haven't done that experiment. We've treated the wild type cells with them, and we don't completely recapitulate just with Gleevec, we don't completely recapitulate the more rapid growth. So the ARCH knockdown cells will grow more rapidly, actually, just in culture.
So
And
if Abel
yeah.
was getting involved,
Right.
of course, that's the prediction.
yeah, right.
You've heard issues. Okay.
Okay, great. David.
I guess this is not. John Condilis has shown that when invasion occurs, you need to have certain isoforms of MENA be present. Have you looked at that, and is the MENA-INV isoform present in that same
Yeah.
Invasive Podia?
We haven't, and it's something, in fact, we have a video conference with them tomorrow. I'll bring it up again. I mean, you know, I've brought it up with both him and Frank, and we talked to him a lot. We haven't looked specifically at it, but it's straightforward to do, and I've offered them samples, so I think it's probably better for them to do it, but yeah.
Lash?
Because just to elaborate on David's point, that isoform seems to be particularly heinous when it appears in terms of promoting invasion. So it's probably also participating in these switches at the tips. Yeah.
So have you had a chance to look at this same phenomenon in other cell lines? So how generalizable this is? Have you looked at other cell lines?
Oh, yeah, okay. Oh, I see what you mean. Yeah, so we've looked in, I should have put that in here. We haven't looked with full ARG knockdown, but we have treated a bunch of different cell lines that would represent, you know, even different, you know, luminal and basal subtypes. And they all have vastly different abilities to invade. So some of them just aren't nearly as invasive. But all of that seems to be, in every case we've tested, the Gleevec seems to tamp down invasion. So, you know, whether that's the same mechanism, whereas Gleevec doesn't seem to have a general inhibitory effect on sort of general migration. So, so we've used that argument, I guess, to our advantage, but we haven't gone in and knocked down it and everything. Human, we've knocked it down in one rat. Very, very invasive rat cell line that people like to use, MTLN3s, and it seems to block invasion there. It seems to inhibit Invatopodia formation.
Okay, time for one last question. Yes, please.
I mean, are ARCH and cortactin expressed in non-invasive tumor cells?
Oh, certainly. I mean, they're in all cells, yeah. And so the question is really, and in fact, part of what my lab's done, that those fibroblasts I told you about, in order to just send out a leading edge and migrate, they need both ARG and cortactin to do that. So I think the question you're asking, if I may be putting words in your mouth, is what sort of funnels all of this into making an invasive podium versus, just regulating regular, just general migration. And we don't know the answer to that. It's very clearly context dependent, and we don't know what the secret is. It could be multifactorial, obviously.
Okay, thank you.
Great, thank you very much.